Class, this is your instructor, Skylar Huff, and welcome back. This is now Chapter 8. It is on mitosis. So, here in this chapter, you're going to learn about the process by which cells divide, and this is asexual division of cells. So, please be sure to review on page 134, including page 135, about tanning, your way to skin cancer. Because though it may be or has become fashionable and has been fashionable for decades, it comes at a price. One price I'll mention is called malignant melanoma. And just remember that indoor tanning is a deadly fad. And this is from your textbook. So let us begin. So the first thing you all will go through is the way in which prokaryotes have a simple cell cycle. So prepare yourselves to diagram the prokaryotic cell cycle, identifying DNA replication, DNA partitioning, and cell fission. So in prokaryotes, the DNA divides first, then the cell divides. So let's go through copying the DNA. All species reproduce, passing their hereditary information on to their offspring. In prokaryotes, the hereditary information, that is, the genes that specify the prokaryote, is encoded in a single circle of DNA called a prokaryotic chromosome. Before the prokaryotic cell divides to reproduce, the DNA circle makes a copy of itself in a process called replication. Starting at one point, the origin of replication, the double helix of DNA begins to unzip, exposing the two strands. So if you look closely at figure 8.1a, it shows how the DNA replicates. So the new double helix that is formed from each naked strand by placing on each exposed nucleotide its complementary nucleotide, that is, adenine with thymine and guanine with cytosine, was discussed back in Chapter 3. Hopefully you remember that process. So DNA replication is discussed in more detail later on, and that's going to be in a chapter called Chapter 11, not later on in this chapter. But when unzipping has gone all the way around the circle, the cell possesses two copies of its hereditary information. So this kind of gives an overview of how DNA replication occurs. So that circular chromosome, beginning at the origin, it unzips, the new double helix is formed, and then from there we have two identical copies. So dividing the new cell, or dividing the cell. So when DNA has been copied, the cell grows, resulting in elongation. The newly replicated DNA molecules are partitioned toward each end of the cell. This partitioning process involves DNA sequences near the origin of replication and results in these sequences being attached to the membrane. When the cell reaches an appropriate size, the prokaryotic cell begins to split into two equal halves, a process called binary fission. So the new plasma membrane and the cell wall are added at a point between where the two DNA copies are partitioned. So if you look again at figure 8.1a on page 136, you will see this, and the same is now shown before you. As the growing plasma membrane pushes inward, the cell is constricted into two, eventually forming two daughter cells. Each cell contains one prokaryotic chromosome that is genetically identical to the parent cell, and each is a complete living cell in its own right. Now let's get to the eukaryotic cell cycle. So here you will describe the phases of the eukaryotic cell cycle, including the three phases of interphase. Cell division in eukaryotes is more complex than in prokaryotes. Eukaryotes contain far more DNA than prokaryotes, and eukaryotic DNA is wound tightly around proteins that condense into a compact shape, the eukaryotic chromosome. The cells of eukaryotic organisms undergo mitosis or meiosis to divide up their DNA. Mitosis is the mechanism of cell division that occurs in an organism's non-reproductive cells, also known as somatic cells. So just keep that in mind. So somatic cells are where mitosis occurs. Meiosis, however, divides the DNA in sexually reproductive germ-line cells. Otherwise, you may hear it referred to as being germ cells. So let's begin the process. So by way of interphase, we have three subdividing phases, G1, S, and G2. So interphase is composed of those three phases. G1 phase, or first gap, is the cell's primary growth phase. For most organisms, this phase occupies much of the cell's lifespan. Next is S phase, which stands for synthesis, or the synthesis phase. The DNA replicates, producing 2 copies of each chromosome. Lastly, here is G2 phase. So during G2 phase, the cell division prepares and begins for that second gap phase with the replication of the mitochondria, chromosome condensation, and the synthesis of the microtubules. So here, let us continue further. So again, the cell is preparing for division. So let's continue on now to page 138 to further the process. So by way of M phase in mitosis, which is continuous but can be thought of as occurring in 4 phases, a microtubular apparatus binds to the chromosomes and moves them apart. And then the 4 phases of mitosis, they are prophase, metaphase, anaphase, and telophase or telophase. And then there is C phase, or cytokinesis. So this is where the cytoplasm divides, creating 2 daughter cells. So if you look closely, you can see that essential biological process. So this is what outlines the process I'm teaching about now, from interphase to mitosis, or M phase. And we'll continue on with the process in just a moment. Chromosomes. So you will distinguish between a homolog and a sister chromatid. Chromosomes were first observed by the German embryologist named Walter Flemming in 1879, while he was examining the rapidly dividing cells of salamander larvae. When Flemming looked at the cells through what would now be a rather primitive light microscope, he saw minute threads within the nuclei that appeared to be dividing lengthwise. So Flemming called their division mitosis, based on the Greek word mitos, meaning thread. Chromosome number. Since their initial discovery, chromosomes have been found in the cells of all eukaryotes examined. Their number may vary enormously from one species to another. A few kinds of organisms, such as the Australian ant, the plant species called Hapapolis gracilis, a relative of the sunflower that grows in North American deserts, and the fungus penicillin, only have 1 pair of chromosomes, while some ferns have more than 500 pairs of chromosomes. So most eukaryotes have between 10 and 50 chromosomes in their body cells, or somatic cells. Homologous chromosomes. Chromosomes exist in somatic cells as pairs called homologous chromosomes, or homologs. Homologous chromosomes carry information about the same traits at the same locations on each chromosome, but the information can vary between homologs, which will be discussed later on in Chapter 10. Cells have 2 of each type of chromosome, and they're called diploid cells. I say again, cells that have 2 of each type of chromosome are called diploid cells. 1 chromosome of each pair is inherited from the mother, and the other inherited from the father. So if you look closely here at figure 8.2, you see to the left-hand side homologous chromosomes. So by way of replication, we now have, again, homologous chromosomes by way of those sister chromatids. Let's continue back where we left off. So chromatids, before cell division, each homologous chromosome replicates, resulting in 2 identical copies called sister chromatids, as shown here to the right. So the sister chromatids remain joined together after replication at a special linkage site called the centromere, the knob-like structure typically observed near the middle of each chromosome. Human body cells have a total of 46 chromosomes, which are actually 23 pairs of homologous chromosomes. In their duplicated state, before mitosis, there are still only 23 pairs of chromosomes, but each chromosome has duplicated and consists of 2 sister chromatids for a total of 92 chromatids. The duplicated sister chromatids can make it confusing to count the number of chromosomes in an organism. But keep in mind that the number of centromeres doesn't increase with replication. So you can always determine the number of chromosomes by simply counting the centromeres. So the centromere can be noticed or seen there in the center of the figure as shown. So next we'll get on to what are called karyotypes. So the 46 human chromosomes can be paired as homologs by comparing size, shape, location of centromeres, and so on. This arrangement of chromosomes is called a karyotype, as shown here before you at the bottom right, and again shown you in your textbook on page 139. Possession of all 46 chromosomes is essential to human survival. Individuals missing even one chromosome, a condition known as monosomy, do not usually survive embryonic development, nor does the human embryo develop properly with an extra copy of any one chromosome, a condition called trisomy. For all but a few of the smallest chromosomes, trisomy is fatal. Even in those cases, serious problems result. Chromosome structure. Chromosomes are composed of chromatin, a complex of DNA and protein. Most are about 40% DNA and 60% protein. A significant amount of RNA is also associated with chromosomes because chromosomes are the sites of RNA synthesis. The DNA of a chromosome is very long. It's a very long, double-stranded fiber that extends unbroken through the entire length of the chromosome. So given that, a typical chromosome contains about 140 million nucleotides in its DNA. Furthermore, if the strand of DNA from a single chromosome were laid out in a straight line, it would be 5 centimeters long. The amount of information in one human chromosome would fill about 2,000 printed books of 1,000 page each. Fitting such a strand into a nucleus is like carrying a string the length of the football field into a baseball that's only one of 46 chromosomes. In the cell, however, the DNA is coiled, allowing it to fit into a much smaller space that would otherwise be possible. So eukaryotic DNA is formed in chromosomes, such as the duplicated sister chromatids seen in figure 8.3 just moments ago. So as we continue on, by winding, and I suppose, figure 8.3 is not shown here, but by winding and twisting the long strands of DNA into much more compact forms. So winding up DNA presents an interesting challenge. Because the phosphate groups of DNA molecules have negative charges, it is impossible to just tightly wind up the DNA. All the negative charges would simply repel one another. As you can see in figure 8.4, the DNA helix wraps around proteins with positive charges called histones. So the positive charges of histones, they counteract the negative charges of DNA so that the complex has no net charge. So every 200 nucleotides, a DNA duplex is coiled around a core of 8 histone proteins, forming a complex known as a nucleosome. The nucleosomes are further coiled into a solenoid. So the solenoid is then organized into looped domains. So the final organization of the chromosome is not known, but it appears to involve further radial looping into rosettes around a pre-existing scaffolding of protein. This complex of DNA and histone proteins, coiled tightly, forms a compact chromosome. Now let's get to the process of cell division. So you will describe the stages of mitosis. When cell division begins in interphase, chromosomes first replicate and then begin to wind up tightly, a process known as condensation. Chromosomes are not usually visible under a microscope during interphase. Interphase is followed by a nuclear division called mitosis. So let's get into the process of mitosis. Although the process of mitosis is continuous, the stages flowing smoothly into one another. For ease of study, mitosis is traditionally subdivided into 4 stages, known as prophase, metaphase, anaphase, and telophase. In prophase of mitosis, the individual condensed chromosomes first become visible with a light microscope. As the replicated chromosomes condense, the cell dismantles the nuclear envelope, and 2 centrioles, or centrosomes in other types of eukaryotes, begin to assemble the apparatus it will use to pull the replicated sister chromatids to opposite ends or to opposite poles of the cell. In the center of an animal cell, the pairs of centrioles separate and move toward opposite poles of the cell, and forming between them as they move apart are a network of protein cables known as spindles or spindle fibers, or the spindle itself. Each spindle fiber is made of microtubules, which are long, hollow tubes of proteins. Plant cells lack centrioles and instead brace the ends of the spindle toward the poles. The spindle fibers attach to the chromosomes, and when the process is complete, one sister chromatid of each pair is attached by microtubules to one pole and the other sister chromatid on the other pole. Next up, metaphase. The second phase to mitosis, metaphase, begins when the chromosomes, each consisting of a pair of sister chromatids, align in the center of the cell along an imaginary plane that divides the cell in half, referred to as the equatorial plane, or otherwise stated as being the metaphase plate. Spindle fibers attach to the centrioles and extend back toward the opposite poles of the cell. In anaphase, the centromeres split and the sister chromatids are freed from each other. So cell division is now simply a matter of reeling in the spindle fibers, dragging the sister chromatids, now referred to as daughter chromosomes, to the poles. Telophase or telophase is where the mitotic spindle disassembles and the nuclear envelope forms around each set of chromosomes while they begin to uncoil. The nucleolus also reappears. So you can see the process here from interphase there at number 1, and this very same essential biological process is also shown to you all on both pages 140 and 141. There is mitosis, showing you what it looks like, and then on the right hand side is step 3 with metaphase, and here is anaphase, there is telophase, and we are now at cytokinesis. So you will contrast cytokinesis in plant cells and animal cells. At the end of telophase or telophase, mitosis is complete. The cell has divided its replicated chromosomes into 2 nuclei, which are positioned at opposite ends of the cell. As mitosis ends, the division of the cytoplasm, called cytokinesis, occurs, and the cell is cleaved into roughly equal halves, the formation of 2 daughter cells, shown at that last panel of the essential biological process 8b, shown here before you, number 6. In animal cells, which lack cell walls, cytokinesis is achieved by pinching the cell into 2 contracting belt of actin filaments. As contraction proceeds, a cleavage furrow becomes evident around the cell circumference, where the cytoplasm is being progressively pinched inward by the decreasing diameter of the actin belt. Plant cells, however, have a rigid cell wall, and those cell walls that are far too strong to be deformed by an actin filament contraction. A different approach to cytokinesis has therefore evolved in plants. Plant cells assemble membrane components in their interior at right angles to the mitotic spindle. So in figure 8.6b, you can see how the membrane is deposited between the daughter cells by vesicles that fuse together. This expanding partition is called a cell plate. So the cell plate grows outward until it reaches the interior of the plasma membrane and fuses with it. So at which point it has effectively divided the cell into 2. Cellulose is then laid down over the new membranes, forming the cell walls of the 2 new cells. So this is figure 8.6 from page 142, mentioned just moments ago. What is cancer? So you explain how mutation is linked to cancer, noting the genes most often involved. Cancer is a growth disorder of cells. It starts when an apparently normal cell begins to divide in an uncontrolled way. The result is a cluster of cells called a tumor that constantly expands in size. The cluster of pink lung cells you see in figure 8.7 have begun to form a malignant tumor called a carcinoma. Shown here to the left in figure 8.7, that is a carcinoma there in the center. So malignant tumors are invasive. Their cells are able to break away from the tumor, enter the bloodstream, and spread to other areas of the body, forming new tumors at distant sites called metastasis or metastases. So that is cancer. So as those cells metastasize or leave and spread, they're going to other areas of the body, forming more tumors, new tumors. Cancer is perhaps the most devastating and deadly disease. Most of us have had a family member or friends affected by the disease. In 2020, over 1 million American men and women were diagnosed with cancer. In that same year, over 500,000 Americans died of cancer. 1 in every 2 Americans born will be diagnosed with some form of cancer during their lifetime. In the United States, the 3 deadliest human cancers are lung cancer, cancer of the colon and rectum, and breast cancer. Lung cancer, responsible for the most cancer deaths, is largely preventable. Most cases result from smoking cigarettes. Colorectal cancers appear to be fostered by the high meat diets favored in the United States. The cause of breast cancer is still a mystery. Mutations cause cancer. And not surprisingly, researchers are expending a great deal of effort to learn the cause of cancer. So scientists have made considerable progress in the last 30 years using molecular biological techniques, and the rough outlines of an understanding are now emerging. We now know that cancer is a gene disorder of somatic tissue in which damaged genes fail to properly control cell growth and division. The cell division cycle is regulated by a sophisticated group of proteins called growth factors. Cancer results from a damage to the genes encoding for these proteins. So damage to DNA, such as damage to these genes, is called a mutation. Cancer can be caused by chemicals that alter DNA, such as the tars in cigarette smoke, by environmental factors such as ultraviolet rays or UV rays that damage DNA, or in some instances by viruses that circumvent the cell's normal growth and division. There are 2 general classes of growth factor genes that are usually involved in cancer: proto-oncogenes and tumor suppressant genes. Genes known as proto-oncogenes encode proteins that stimulate cell division. So mutations that activate or improve the functioning of these genes step on the accelerator, so to speak, of cell division, causing mutated cells to divide excessively. So mutated proto-oncogenes become cancer-causing oncogenes. Releasing the brakes, or the tumor-suppressing genes, are the second cancer-causing gene. So again, tumor-suppressing genes. Cell division is normally turned off in healthy cells by proteins that are encoded in tumor-suppressing genes. Mutations to these genes essentially release the brakes of cell division, allowing the cell containing the mutated gene to divide uncontrolled. And that mutated gene is called TP53. That is the name of the gene. So the cell cycle never stops in a cancerous line of cells because that tumor-suppressing gene, TP53, now has a variant or mutation. So, next up, let's get to just a bit more information about the nature of lung cancer. So, of all human diseases, none is more feared than cancer. Nearly 1 in every 6 deaths in the United States was caused by cancer in 2020. The American Cancer Society estimates that 606,000 1,520 people died of cancer in the United States in 2020. About 22% of these, 135,720 people, died of lung cancer. And then about 135,000 cases of lung cancer are diagnosed each year, and 90% of the persons die within 3 years. So the cause of lung cancer, let's get to it. So the search for a cause of cancers, such as lung cancer, has uncovered a host of environmental factors that appear to be associated with cancer. For example, the incidence of cancer per 1,000 people is not uniform throughout the United States. Rather, it's centered in cities such as the heavily populated Northeast, indicated by the red areas in figure 8.10 on page 146. So, given that, it's also going to be found in the Mississippi Delta, indicated by the red and brown areas, suggesting that environmental factors such as pollution and pesticide runoff may contribute to cancer. So when the many environmental factors associated with cancer are analyzed, a clear pattern emerges. Most cancer-causing agents, or carcinogens, share the property of being potent mutagens. So a mutagen is a chemical that damages DNA, destroying or changing genes. So the cancer is caused by a mutation that is now supported by an overwhelming body of evidence. So what sort of genes are being mutated? In the last several years, researchers have found that mutation of only a few genes is all that is needed to transform normally dividing cells into cancerous ones. In identifying and isolating these cancer-causing genes, investigators have learned that all are involved with regulating the cell proliferation. So a key element in this regulation are tumor suppressors, or genes that actively prevent tumors from forming. So how do tumor suppressor genes work? So TP53 protein, a tumor suppressor I mentioned earlier, sometimes called the guardian angel of the cell, inspects the DNA before the cell divides. When TP53 detects damaged or foreign DNA, it stops cell division and activates the cell's DNA repair systems. If the damage isn't repaired in a certain amount of time, TP53 pulls a plug, triggering the events that killed the cell. This is known as apoptosis, or programmed cell death. So in this way, mutations such as those that cause cancer are either repaired or the cells containing them are eliminated. So if the gene that produces the TP53 protein itself is destroyed by mutation, the future damage accumulates unrepaired, including mutations that lead to cancer. So fully, 50% of all cancers have a disabled TP53. If cancer is caused by damage to growth-regulating genes, what then has led to the rapid increase in lung cancer? Two lines of evidence are particularly telling. The first consists of detailed information about cancer rates among smokers. The annual incidence of lung cancer among non-smokers is only a few per 100,000, but it increases with the number of cigarettes smoked per day to a staggering 300 per 100,000 for 30 cigarettes a day smokers. A second line of evidence consists of changes in the incidence of lung cancer that mirrors in smoking habits. So that mirrors those changes, in smoking habits. So look carefully at the data presented in figure 8.11 on page 147. The upper graph is compiled from data on men and shows the incidence of smoking in the blue line and of lung cancer, the red line. And this was in the United States in 1900. As late as 1920, lung cancer was a rare disease. But about 30 years after the incidence of smoking began to increase among men, lung cancer also started to become more common. Now look at the lower graph, which presents data on women. Because of social mores, a significant number of women in the United States did not smoke until after World War II. See the blue line shown there. So when many social conventions changed. So as late as 1963, only about 6,588 women had died of lung cancer. But as women's frequency of smoking has increased, so has their incidence of lung cancer, the red line. So again, with a lag of about 30 years, women today have achieved equality with men in the number of cigarettes they smoke, and their lung cancer death rates are now rapidly approaching those for men. So in 2014, 72,330 American women died of lung cancer. How does smoking cause cancer? So cigarette smoke contains many powerful mutagens, among them benzoaprine, and smoking introduces these mutagens to lung tissues. So benzoaprine, for example, binds to 3 sites on the TP53 gene, and it causes mutations at these sites that inactivate a gene. And it was in 1997, scientists studying this tumor suppressor gene demonstrated by a direct link between cigarettes and lung cancer. So they found that TP53 gene is inactivated in 70% of all lung cancers. So when inactivated, TP53 genes are examined, and they prove to have mutations that at just the 3 sites where benzoaprine binds. So clearly the presence of the chemical benzoaprine in cigarette smoke was responsible for the lung cancer. So the most effective way to avoid lung cancer is to not smoke. On a statistical basis, smoking a single cigarette lowers your life expectancy 10.7 minutes. That's more than the time it takes to smoke a cigarette, you all. So every pack of 20 cigarettes bears an unwritten label. The price of smoking this cigarette pack is 3.5 hours of your life. So smoking cigarettes is very much like going into a totally dark room with a person, and this is in your textbook. I repeat, this is in your textbook, with a gun and standing still. The person with the gun cannot see you and does not know where you are and shoots once in a random direction. A hit is unlikely, and most shots miss. As the person keeps shooting, however, the chance of eventually scoring a hit becomes more and more likely. And this is in your textbook. This is in your textbook. I am not saying this. So similarly, every time an individual smokes a cigarette, mutagens are being shot at his or her genes. The more cigarettes smoked, the more likely the genes will be damaged. So as I end things, it states that in 2020, 13.5% of Americans were cigarette smokers. So in an attempt to lower that number, the federal government has proposed new regulations strictly limiting the amount of nicotine in cigarettes to non-addictive levels. So this has been your instructor, Scholar Huff. This has been chapter what is called 8 on the cell cycle. And please keep in mind that you should review the Putting the Chapter to Work on page 150, and also review the Retracing the Learning Path, found that very same place. Thank you for listening, and prepare well for your test.